Hey everybody! Today we're going to be talking about the MacConkey auger plate that we're going to be using for our lab. Now, the MacConkey auger plate is both a selective and differential medium generally used to look for gram-negative enteric bacteria and especially sort of break them down based on their ability to ferment lactose. So our medium has the pH indicator neutral red in it, and neutral red will be colorless at pHs 6.8 and above. But when you go below pH 6.8, it's going to start turning red. So in terms of the media basics for our medium here, basically in terms of the selected component, our medium has both biosalts and crystal violet in it. And both of these guys are pretty good at killing off gram-positive bacteria, and that then helps us select for the gram-negative bacteria that are going to be left. In terms of the differential component, we have lactose in the medium as well. And basically, if an organism or if a bacterium is able to break down lactose or ferment it as a food source, then it's going to create acidic byproducts, and those acidic byproducts will decrease the pH of the medium, and we're going to start seeing red colorations. So with that in mind, let's take a look at our results and how we can basically score our plate. So for this plate here, this was a mixed culture that was put on the plate, and because of that, we got a couple positives and some negatives on here. So if we get these kind of yellowy, translucent, or white colonies on our plate, those are going to be negative for lactose fermentation. However, any of these guys that produces kind of these red centers or just red colonies altogether are going to be positive for lactose fermentation. Now, in terms of inoculating our plate, we can do a standard broth to plate transfer. That's done most commonly. We can also do a plate to plate or slant to plate if you have your sample on a plate or a slant. Or what we might even do for some of our labs, if we're doing a dilution lab, sometimes we might also be putting milk on there or water samples on there, and at that point we would be using our pipettes, the little green pipettes, in order to do a transfers that way. So a couple different ways that we can inoculate this plate here. But that should really cover everything that we need to know for our MacConkey auger plate.